Good morning friends. Today we learn about TS of liver from BSc third year class. Paper number 2, mammalian histology. Liver is the largest digestive gland in the human being. This liver is largest exocrine gland. This mainly secretes bile secretion, and these are again compound tubular serous gland located below the diaphragm above the intestine. And functions of liver is exocrine gland and mainly secretes bile. Liver mainly secretes bile. Second function, synthesis of prothrombin. Synthesis of prothrombin, and this prothrombin is useful in blood coagulation. Liver storage glycogen, fats, proteins and vitamins A, D and K. Vitamin is stored in the liver. This liver also excretes waste products and detoxifies various metabolites. Liver detoxifies various metabolites. When we observe microscopic structure of liver, mainly these livers consist hepatic lobules. This microscopic structure forms 3 components. First component is parenchyma, second component is stroma, and third component is blood channels. This first component, parenchyma, mainly consists hepatic cells, or it is called as hepatocytes. These are structural and functional unit of the liver, and these, when we observe this histological slide, this liver is incompletely covered by an outer tunica serosa derived from the peritoneum. Liver is covered by connective tissue. It is called as a Gleason's capsule, and these Gleason's capsule present only in portal area. These Gleason capsules only present in this portal area, and these capsules contains abundant elastic fibers present. First histological part is hepatic lobules. Hepatic cells are arranged into minute irregular polyhedral blocks called as liver lobules or hepatic lobules. These are arranged irregularly polyhedral blocks, and it is called as hepatic lobule or liver lobule. Each lobule in human being, these are about 2 mm tall and 1 mm wide. Each hepatic lobule in human 2 mm tall and 1 mm wide. The lobules are most distinct in pig, rat or cat. The spaces formed between neighboring lobules are filled with connecting tissue. In between this hepatic lobule, this space is connected with connective tissue, filled with connective tissue, and in between this connective tissue here find bile canaliculi is present in between this connective tissue. Find bile canaliculi is present. Next cell is hepatic cells. Here I am drawing two hepatic cells. These hepatic cells having centrally located nucleus. Nucleus may be 2 or more in number, and these liver mainly component is hepatic cells, and these parenchyma mainly consist the hepatic cells. These hepatic cells are polyhedral in shape, and the cells are distinct cell membrane. Each cell has centrally placed nucleus. These nucleus may be 2 or more nucleus present. Cytoplasm contains numerous mitochondria, Golgi complex. In single cell also 50 Golgi complex is present. In each cell 50 Golgi complex present, and this cytoplasm also contains glycogen and fat droplets are present. The hepatic cells are arranged irregularly. Hepatic cells are arranged in irregular branching interconnected plates which appears like cord-like structure. These hepatic cells are arranged a radiating manner and forms hepatic cord-like structure, so it is called as hepatic cords. These are called as hepatic cords, and the plates or cords are called as hepatic cords. The plates radiating manners, these plates are arranged in radiating manner around the central blood vessel of these lobules. These plates are arranged radiating manner towards the periphery around the central vein, and these in between these hepatic cells fine canaliculi, bile canaliculi is present. Next, hepatic sinusoids. In between the hepatic cells, here space present, and this space is called as sinusoids. These sinusoids make up the intralobular system of blood capillaries which convey the blood from the portal areas to the central vein. Here these hexagonal cells, here central, in the central vein is present, and from the portal area here space is present, and this space is called as sinusoid. In this sinusoid, here hepatic artery and hepatic veins enter, and this blood flow towards the central vein. Blood flow towards the central vein. It is called as centrifugal force. This blood collected in the central vein, and these sinusoids receives blood from the branches of hepatic portal vein and branches of hepatic portal artery. These sinusoids are lined by endothelial cells like present in the blood vessels. At the end of sinusoid, there is sphincter to control the flow of blood. in this sinusoid, here endothelial cells are present and these endothelial cells are fenestrated, and from this blood is passed towards blood plasma pass towards the hepatic cells, and from this blood plasma here again Kupper cells are present. Here Kupper cells are present. These Kupper cells are phagocytic in nature. Here phagocytic in nature, and Kupper cells nucleus is oval nucleus. These Kupper cells involved in breakdown of RBCs, fat metabolism, and in the formation of bile pigments. These old Kupper cells gives new Kupper cells are formed from the pre-existing Kupper cells. Next part is portal area. These sinusoids are made up of endothelial cells which are fenestrated, and these sinusoids again lined Kupper cells. Kupper cells are phagocytic in nature, involved in breakdown of RBCs, involved in fat metabolism, as well as these Kupper cells produce new cells. Next point is perisynoid space. It is also called as space of Dissay. The space between sinusoid, here perisynoid space of Dissay is present. Space of Dissay, this space between here hepatocytes and sinusoids, here space of perisynoid space, it is also called as space of Dissay present. Here in this space of Dissay, fat storing cells, it is also called as lipocyte cells. Fat storing cells, lipocyte cells are present, and these space stores the fat droplets. Next point in our list is bile duct. Here bile duct, it is also called as hepatic duct, and this hepatic duct present in between the minute channels, it is called as bile canaliculi in between hepatic cords. Here small duct present, it is called as bile canaliculi. This bile canaliculi opens into smaller bile duct. These are lined by cuboidal cells, and these small duct join the large duct which are lined by columnar cells. All large ducts here liver present, and here large duct from right side and large duct from left side joins to forms common hepatic duct. Here gallbladder present, and gallbladder carries cystic duct. This cystic duct also joins bile duct and forms common bile duct. This common bile duct opens in the duodenum, and before open into duodenum, here pancreatic duct join and forms common hepatopancreatic duct, and this hepatopancreatic duct open into the duodenum. Next part, blood supply. Liver receives arterial blood from hepatic artery. Liver receives blood from hepatic artery and venous blood from the hepatic portal vein. The veins brings blood from intestine. Here hepatic artery and hepatic vein. Hepatic vein carries venous blood from the hepatic portal vein, and this hepatic portal vein about 75% blood carries, which is less oxygenated, and this carries from the intestine, which is laden with the carbohydrates, fats and proteins. The liver cells stores this substance in their cytoplasm. Hepatic veins, these hepatic veins drains the blood into the inferior vena cava. Hepatic artery carries blood towards the liver about 25%, and it is more oxygenated blood. Diagnostic characters of the liver. Liver distinguished from other organs such as Gilson capsule is present in livers, as well as numerous hepatic lobules are present in the center of lobules. In the center of lobules, here central vein is present. Hepatic cords are also present. These hepatic cells are arranged in radiating manners like cords. It is called as hepatic cords. These hepatic cords are present in liver. Sinusoids and portal areas are present in between hepatic sacs. Here space present, it is called as sinusoids, and hepatic area present. These hepatic area mainly consist hepatic artery, bile duct and hepatic vein. It is also called as hepatic triads in early histology. Here hepatic artery, bile duct and hepatic veins present, but later on it again discover. Here his small plasma remains, and this plasma converted into lymph, and here this hepatic triad lymph vessel is also present, and it is also called as tetrad, but it is commonly called as a portal area, and such portal area is present in the liver. This is all about the histology of liver. Thank you.